All right, this is part 2, starting at 17.3. So the mRNA has been created, and now we have to, in eukaryotes specifically, we have to modify the pre-mRNA before sending it off to the cytoplasm where the ribosomes are. So here's how we do it. First off, at the 5' end of that messenger RNA, we attach a 5' cap, or a G cap, as it's called. It uses guanine. It's added by GTP. You can see here a G for guanine and 3 phosphate groups. This is added to the 5' end. The 3' end gets a poly-A tail, which is many AAAs, as you can tell, a polyadenylation signal. And these two serve to protect the ends to both hydrolysis and to new things binding to them to potentially alter their chemical bonding interaction type stuff. So again, poly-A tail on the 3' end and 5' cap on the 5' end, a G cap on the 5' end. We also have to remove introns. Now, genes are made of introns and exons, and the easy way to think about this is still in the nucleus. Okay, here's RNA processing in the nucleus. These introns are going to stay in the nucleus, and the exons are going to exit the nucleus. So we need to cut the introns out, leaving only the exons behind, and we're going to splice together the exons, because the exons are the part that we're actually going to read to code into a protein. This is typically carried out by spliceosomes, a really fun name, which is a combination of some RNA and some other proteins, and they help to fold in the introns so that the two corners of the exons meet, and then they pop out or cut out this segment of the intron, and then that intron will get recycled, leaving behind only the exons there. Some organisms use ribozymes more, which again is catalytic RNA that helps to splice its own RNA and remove the introns. But not all of them do, and this is just a reminder that not all biological catalysts are actually proteins or enzymes. They can also be RNA. Now, one interesting thing that thus affects the one gene, one enzyme hypothesis is this idea of alternative RNA splicing. If I go back a couple slides to here, because these exons are the only things that are kept and taken to the ribosome, if the cell considers at every moment introns in different places, or introns don't always have to be here. This could be an intron, or this could be an intron, or this could be an intron. And that's going to alter how the DNA gets cut, and thus which exons are left behind. Thus that one gene can have many different mature mRNA sequences, and thus code for many different proteins. That's what we see here for alternative RNA splicing. Which segment is treated as an exon can actually vary, and thus one gene or one gene segment can give rise to many different polypeptide chains. And we also do know as well that typically different exons code for different domains or different regions of the protein. So here's exon 1, exon 2, exon 3. Cut out the 2 introns, and this exon 2 codes for this domain, exon 1 codes for this domain, and then all together we have this polypeptide. So they're each different segments of the polypeptide. Now we get into translation. This is the overall basic idea, just to give you the big picture, converting mRNA into protein. This is a useful animation if you want to click on it from the real actual slideshow. Here we have tRNA, and to that tRNA we have an attached amino acid in purple at the top. At the bottom we have an anticodon. That's actually labeled here. The anticodon will bind to the codon. Now when you look up in the chart, once again, you look at the codon. This anticodon just happens to bind to the codon to hold it in place, giving time for the amino acids to create a peptide bond between the two of them. And then you have this elongating polypeptide, that amino acid strand. Here in brown you see the ribosome. At the top is the large subunit. The bottom is the small subunit. Now let's get into the details. Not all tRNAs are identical. At one end I said we have an amino acid. At the other end is an anticodon. And here's basically what it looks like. Here's the amino acid binding site. Notice it's the 3' end here of this messenger RNA. Not messenger RNA, this tRNA, which got folded up into this funny shape. I wonder why they call it a T. Besides transfer, it kind of looks like a T if you ask me. And here's the anticodon that got folded around, AAG, which will bind to, you can guess, UUC. Again, it's U, not T, of course, because this is RNA. This is about 80 nucleotides long. That's not that important. Here you see an actual real picture of it. Here's the tRNA, anticodon. This is what it looks like, kind of how it folds in reality, three-dimensionally. Now, the enzyme that helps to bind the amino acid to the tRNA is called aminoacyl-tRNA synthetase. And here we can see through the help of ATP, we have the amino acid entering this enzyme, aminoacyl-tRNA synthetase. You might want to say that to yourself once, a strange name. I paused to let you say it to yourself, but if you didn't, you can go ahead now. Phosphate groups are cleaved off. tRNA enters the enzyme as well into the active site here, and a bond is initiated by hydrolyzing that phosphate group, providing energy to shift the enzyme, and a bond is created between the amino acid and the tRNA, and then the tRNA pops off. And here's the aminoacyl-tRNA. That's the activated amino acid. Ribosomes help to then take the tRNA and attach its anticodon to the codon to allow the amino acids to bond together. Here we have large subunit on the top, small subunit on the bottom, with 3 regions in the large subunit for binding and so on. Now, ribosomes are made up largely of protein and rRNA, or ribosomal RNA. Here's the 3 sites. We have the E site or exit. Sometimes it's pictured or shown on the other site, depending on how you're looking at it. P in the middle and A on the other side, opposite the E. These details, these letters, not important. Just know it has some entry sites where the tRNAs will enter. And so here we see tRNA entering. We have one tRNA with its amino acid chain growing. This is already partway down the line, the growing polypeptide. A new one comes in to the A site, as you can see here, with its amino acid. A bond will be created between the two. What's this slide? So here, a bond will be created between the two, and then this entire polypeptide chain will now be attached to this tRNA. This tRNA will shift over to the P site. This one will now have no amino acids attached to it. It'll shift to the E site, as you see here coming off, and a new one will enter into the A site to continue the process. Now, again, this begins with initiation, elongation, and termination. And here we see initiation. The first tRNA always carries methionine, or MET, because AUG, the start codon, codes for methionine. Now, methionine is not necessarily always the first amino acid in a protein. It could be cleaved off or cut off as folding and processing occurs. But in general, it always begins with MET. MET binds to the start codon. Large subunit gets added on, and here we see it in the P site, ready to go. Initiation has occurred. Then you get a new one adding, as we saw before. A bond is created between the two. That is a peptide bond that requires energy in the form of GTP to do so. Then you see the amino acid chain, this peptide chain, polypeptide, has shifted over onto the A site. The amino acid that was attached to here is now right here, just to give you an idea. And this ribosome will now shift down one codon to your right, essentially. This one will then be in the E site. This one from the A will go into the P, as you see here, and now leaving space for a new one to enter into the A site, as is occurring there. This whole process finishes, translation stops, when a stop codon is read in the mRNA, and a release factor hydrolyzes the bond between the tRNA and the P site. So here you can see this release factor, which is coded for by the stop codon. You don't have to memorize these because they'll be in the chart, as you saw previously. And then that basically explodes the whole ribosome, and it causes everything to fall apart, and the polypeptide is done. Here you see the real thing happening. This process can occur simultaneously. This is actually in a prokaryotic cell being shown, but it can happen in a eukaryotic cell. We have multiple ribosomes bound to the same messenger RNA, making the same protein over and over and over very, very quickly. Here you see an actual electron micrograph, scanning micrograph picture of that. And then these polypeptide chains have to undergo modifications. They have to form their three-dimensional shape to have activity, as you know. Secondary structure, tertiary structure, and quaternary structure in the end. And you may remember chaperonins are these assisting proteins. They're kind of like buckets that will take the other protein and put it inside the chaperonin and assist in the folding by creating a perfect microenvironment around the protein needed to be folded. And then you open up the bucket and the actual folded protein comes out of the chaperonin. Now there's two types of ribosomes. There's free ribosomes and there's bound ribosomes. Bound meaning bound to the endoplasmic reticulum. First, to start off, free ribosomes are found in the cytosol, and they initiate the synthesis of all proteins. Every single protein, whether it's going to be in the ER or going to be in the cytoplasm, starts on free ribosomes floating around in the cytoplasm. Now, if the ribosome, once initiation has begun, if the ribosome does not attach to the endoplasmic reticulum, then the protein will stay in the cytoplasm, meaning the protein that gets made by this ribosome is for this cell. If it does attach, well, then it's entering into the endomembrane system, and eventually it's destined for secretion. It's going to find its way outside the cell or into the membrane of the ER or Golgi. And how do ribosomes know whether to bind or not? Well, on the end of the protein being produced, there's something called a signal peptide, and this signal peptide gets this signal recognition particle, or SRP for short, that binds to it. And when the SRP binds to it, that signals it or enables its attachment to the ER, and we can see that here. Here's the signal peptide in green. The SRP binds to it and initiates its attachment to this SRP receptor in the membrane of the ER. So here's the lumen of the ER, here's the membrane. This ribosome will bind, and then as the the polypeptide is being synthesized, it's getting synthesized into the ER, and then once it's removed, it's now inside the endomembrane system, destined for eventually the Golgi, and then outside the cell. So some quick review. RNA can hydrogen bond to other nucleic acid molecules. It shares the same principles, same basics as DNA. And we usually find RNA single-stranded, but you can find it double-stranded as well. It can have a specific three-dimensional shape, and you do know that it can act as a catalyst in the form of a ribozyme. There's multiple types of RNA. We don't have to memorize all the different types right now. Messenger RNA, tRNA, and rRNA we talked about largely today. Some snRNA we talked about briefly, although we didn't mention it as snRNA. srpRNA, signal recognition particle, we talked about. And then some of these other ones, which we're not going to talk about. Now, prokaryotic cells, they don't have a nuclear envelope, so as we mentioned before, while transcription is occurring, translation can begin all at the same time, and you can see that here as this mRNA in red is being produced, ribosomes attach and begin translation, also because we don't need to modify this pre-mRNA. Prokaryotes don't use introns, and we don't need a 5' cap and poly A tail and all that good stuff. Whereas we did in a nucleus, in a eukaryotic cell, the nuclear envelope separated them, and then we have to do that RNA processing. We'll take a pause on this one.